Hello, everyone. I'm Annie Dean, and I'm one of the co-founders of the Kabuki Syndrome. I'm pleased to introduce our next segment. Quick housekeeping item that this meeting is being recorded, and by continuing to be in the meeting, you are consenting to be recorded. Continued thank you to our sponsors, the Knights of Columbus, our new sponsor, the Crossman family, All Stripes, HealX, and the Lily Ingram family. I am so excited to introduce to you two amazing Kabuki Syndrome advocates, Jennifer Manti-Goldsmith, proud Kabuki parent to Hutch, and Dr. Kasturi Haldar. One of the most incredible things about being part of the Kabuki Syndrome Foundation is being able to connect with amazing families like Jennifer's and see the incredible research that they've been able to seed and support through partnerships like Notre Dame, and to learn about incredible work like Dr. Haldar's work. Jennifer is going to give us a quick overview and discussion of a section called Supporting Kabuki Syndrome Research Through a Patient Family Perspective. And then we'll transition into Dr. Haldar, whose preclinical studies on long-term treatments for cognitive functions in a Kabuki syndrome mouse model will be very interesting to hear. After we hear from Jennifer and Dr. Haldar, then we will make sure to transition into our research roundtable. So please share your questions as soon as that section ends. And it sounds like we are going to transition directly to Dr. Haldar first. Thank you so much.
Well, I'm Kasturi Haldar, and I work at the University of Notre Dame. I direct the Center for Rare and Neglected Diseases there, and Jennifer isn't here, but I'll introduce Jennifer. So this is how we got introduced to Kabuki Syndrome. So Jennifer's, you can see here, Jennifer, her family, her husband, Spencer Goldsmith, her children, Ellie and Hutch Goldsmith, and her parents, James Manti and Sandra Manti. And they were the ones who came to us asking whether we would be interested in developing a program with in Kabuki Syndrome. And the funding has come from Mr. and Mrs. James Manti, other families with Kabuki Syndrome, and then their families and friends. And we're very grateful because we probably would not have been able to work in this area had it not been for them getting us started. So this is a dated picture where they're all visiting the mouse lab. You can see they're all booted up. I did have the opportunity to see Ellie and Hutch recently. They've really shot up. But the context was a very sad one, which is that Jim Manti passed away last month. And I just wanted to take a minute to honor his memory. In addition to his professional success and his very strong ties to his own family as well as the Notre Dame family, he was really very interested in trying to understand the causes of intellectual disability in a broad and general way. And so he also had a great sense of humor, and we will all really miss him because he was a prime mover of the work. So he came to see me because my lab has been interested for a while in how HDAC inhibitors remodel the mammalian brain. So HDAC inhibitors are approved as therapies for rare cancers. They're also of interest in the treatment of neurodegenerative disorders, but brain function requires HDAC activity. So there's a nub there. And the other issue is that neurological disorders potentially require lifelong treatments, as opposed to in a cancer, when you have the tumor, you might take a drug that makes you feel not so good, but once the tumor resolves, you get off the drug. And that's the way a lot of chemotherapy works, but that's obviously not going to work in a neurological disease. So the challenges are that HDAC-I therapy will have broad effects on transcription because they're changing the state of chromatin. And this is going to lead to changes in expression of hundreds of genes. which can be quite dangerous. So you really need to be able to keep the dose low. And you need a therapeutic strategy that not only lowers the dose, but allows penetration across the blood-brain barrier and sufficient brain function for HDAC function, right? You can't turn those HDACs off completely. And so what we were interested in asking is, could we do this with an FDA-approved HDAC inhibitor? And why FDA-approved? Well, the sad reality is that a new chemical entity, even if a major pharmaceutical was undertaking it, would take 10 to 15 years to go to the clinic. So you can cut a lot of that time if you can cut short the regulatory and tox efforts that an FDA-approved drug has already been through. And you also can't go from no drug to the perfect drug. So sometimes going the route of using something that is advanced enough and adapting that to optimize the treatment is a more productive way, especially in the space of rare diseases where the unmet need is very high. We took an approach which is essentially a pharmacological approach of improving the plasma exposure of a drug using a formulation. And in this case, we took the FDA-approved HDAC inhibitor, Vernistat. It was the only approved inhibitor when we first started, hydroxypropyl beta-cyclodextrin, as well as polyethylene glycol. And what you can do with this is essentially make a complex, as shown on the right-hand side of the screen, inject that into the bloodstream, where this three-component complex now starts to come apart and release components slowly into the bloodstream. So you can just think about it as a slow release of a more soluble form over a longer period of time, which reduces toxicity, increases the net amount available to cross the blood-brain barrier and effect the change that you're looking for. So the notable thing was that we did the screen directly in a mouse. So why a mouse? Well, they're mammals. We are mammals. Looking at, even if you don't have the indicator of which is wild type and even pig type C, so we essentially applied these formulations directly to NPC mice. And the idea was to really try to go from what you see on the right-hand side to the left-hand side. Mice showed neurobehavioral manifestations that are very easy to recognize. And anybody who has done a behavioral assay will recognize that, mice can smell your perfume. Mice are also, if you try to work with larger animals in the laboratory, you just cannot manipulate them, mammals in the laboratory, as well you can mice. And that's really why there are so many reagents in mice. So the third component that's a little different is we took a stepwise delivery of a very small amount of drug into the brain to enable long-term, what we call tolerable remodeling of gene expression in the mouse brain. So if you think about it, if you have wild-type mice and you inject something that changes transcription, you will get a spike. And then because of the homeostatic response, that spike will actually come down. And you go back to the resting state. You can think about it like drinking a cup of coffee. It's pretty similar. You get a change, you get a little bit of a high, and then it comes down. And then you can drink some more, and you get a high, and it comes down. But when you have a mutation, you've lost that homeostatic response. And so you actually can get the transcriptional change in response to your perturbant, but you don't come back to steady state. Instead, you come back to an intermediate state, and then you can get another spike, which will take you to a second, once it's allowed to relax, to a second intermediate state. And gradually, you can go to what we could call from step 1 to step 2, where you're getting remodeling, but you may not be getting any functional benefit. And then finally, to a desired state of remodeling, where you do get functional benefit. And if you try to go directly from step 1 to step 3 with very large amplitude, that rapid transition creates an intolerable inflammation. So that was our strategy, and we ended up being successful at it. And so here's the paper that was published in 2016, where we showed that you could chronically administer an HDAC inhibitor to treat both neurological and systemic Niemann-Pick type C disease in a mouse model. And this was really the first demonstration. The paper received a lot of attention, if I say so myself. It's still actually in the top 5% of all altmetric outputs of publications. And it received the attention because really the first demonstration of an HDAC-I remodeling of gene expression in mammalian brain with long-term neurological benefit and survival, i.e. not only did we improve the tissue in the brain, but we actually improved the entire state of the mouse. And it emerged from a 2-year screen in mice, which is longer than in some other systems, but it didn't take an army. It was one talented postdoc, Suhail Alam, a very talented technician, Michelle Getz. So it actually can very much be done in the context of an academic lab. So in addition to the efficacy studies, we then undertook safety studies, of course, in the mice. And both were pretty extensive, and we were able to establish that this chronic administration had no deleterious effects for neurological, visceral, lung, or brain HDAC expression, because that was obviously a big concern. There are many HDACs that are essential for brain function. And this was over 10 months of weekly administration. One of the reasons we stopped is because Michelle Getz sort of said, look, these mice are doing just fine, and I'm tired of injecting them. So we said, all right, time to stop, let's write it up. So in Niemann-Pick, the cerebellum is a very important part of the brain. And so this is just data showing that, you get preservation of Purkinje neurons, those are the biggest neurons. And you can see the green color in the untreated healthy animals versus diseased animals, and then Niemann-Pick with TCF. And basically, we're able to put back a quarter of the Purkinje neurons. So that's really a substantial portion of the neurons. I suspect most of us don't use more than a quarter of our neurons, even on a good day. So we, in Kabuki syndrome, of course, focused on the hippocampus, which I think you all know, but I'll just quickly go through, is a small curved formation in the brain. There are 2 hippocampi, one on each side of the human head. The hippocampus controls memory, learning, emotion, nociception, and importantly, it is where new neurons can be generated in the adult brain. And so hippocampal dysfunction in Kabuki syndrome, the hypothesis is affects learning, memory, and mechanosensory responses. Okay, so Hans Bjornson had published a very nice paper in Science Translational Medicine, which I know is well known to all of you, showing that AR45, an HDAC inhibitor, could in fact promote neurogenesis. So we obtained the same model, back-crossed it in our lab, and then proceeded with weekly injections of TCF, starting from post-weaning, 90, and then up to 100 days. And to look at postnatal formation of neurons in the hippocampus, you have to look in a particular region called the subgranular zone of the dentate gyrus. So the gray outlines that you see are the granule neurons. And what you see in terms of cells depicted in green are the different steps that postnatal neural progenitor cells have to undergo in the SGZ to finally become mature neurons. And since the hypothesis is the stimulation of postnatal neurogenesis reduces intellectual disability in KS, when you're looking for, have you actually achieved the stimulation of postnatal neurogenesis, one of the main markers that you use is for the neuroblast stage, because it says that the cells have progressed all through, and this is what the DCX staining is about. There are other ways of assessing it, and Dr. Bjornson elucidated some. We use all of those assessments. And to evaluate a triple combination formulation, just to cut it short, you have to evaluate the TCF, but you also have to evaluate every component individually and in combination. And as a consequence of that, we actually found that we have 2 effective drug formulations in the mouse model. Treatment 1 stimulates postnatal neurogenesis in the dentate gyrus and restores hippocampal cognitive functions, which is what you would expect. But treatment 2 does not stimulate postnatal neurogenesis, but it still restores hippocampal cognitive functions. So we decided that to mechanistically differentiate between T1 and T2, that we needed to map the hippocampus. And so to do this in an unbiased way and to understand the effect of each treatment on all cells, we undertook single-cell RNA-seq of the hippocampus. And so that's laid out, I think, fairly clearly here. You dissect the hippocampus, you then disrupt the hippocampi, you prepare and collect all single cells, and then you sequence every cell. You get the transcriptional profile of every single cell because they can be different even if they're coming from the same tissue. And that's just shown schematically on the right-hand side here. Gene 1, for instance, might be in 150 copies in cell 1, but only in 73 copies in cell 2, et cetera, et cetera. So once you get all of this very detailed information on every single cell that you isolated, you then integrate it computationally and you view the output. What do you get? So you get a map. What you're looking at here is a map of the hippocampus. And this is input from Kabuki mice, from wild-type mice, treatment arm 1, treatment arm 2. There's 60 mice in here. It actually took several months to do the experiment because you can't actually handle that many mice at one time. And there's several million data points. Each of the columns are clusters, and there are 28 different clusters, one per column, isolated from the hippocampus. And what can you interpret from this? Well, one of the things is this is a cluster proportion map. And so it tells you the extent to which a treatment affects the cell type. And you can sum these responses to tell you the overall response of the hippocampus. So what you see for treatment 1, which is shown in red in the key on the right-hand side, is that it has a very restricted response, suggesting that its stimulation of postnatal neurogenesis comes with very few other effects across all hippocampal cell clusters. And that is very good news because we want to be able to stimulate the processes we're interested in stimulating in the brain, but we don't really want to create a lot of non-specific or other stimulations. If you look at treatment 2, which is the dark hunter green, you can see that that has a larger transcriptional amplitude than treatment 1. And what that tells you is really right away that treatment 2 uses a very different mechanism from treatment 1. And we are now going through the analysis of mechanisms through what is called trajectory analysis. So what you can do is, a time series of looking at the difference in hippocampal cells as they transition from the untreated state to the treated states. And this is a visualization of a simple theoretical example of trajectory for effector cells, let's say in treatment 1. And I emphasize we're still going through the trajectory analysis, but I just wanted to show you how the data analysis is done. So you can look at untreated Kabuki cells, for instance, at animals that are at 1 month, and you can see how that progression goes from 1 month to 3 months. What happens if they are mock treated? Because for any treatment, you always have to have the corresponding mock treatment. And then finally, what are the cells that actually emerge in the treatment 1 effector population? And that's how you can tell what is the mechanism here? What cell types are you inducing? And then you can dive down into them because, they come with their complete genome sequenced. So you get a very good idea at the cellular and the molecular level as to what is making your behavior change occur. So I'd like to wrap up and just, again, thank the families, the Manti family, the Manti-Goldsmith family, as well as other Kabuki syndrome families who got us started. They're really remarkable people. And, we would have, as I said, not done this work had it not been for them. The people in the lab who are presently working on it, Ryan McCardle, who is a grad research assistant, and Prasad Padmanabhan, who is a research associate, and they're really pretty remarkable for what they can do with the mouse hippocampus, as well as the computational analysis that's required to do the single cell RNA-seq work. And they work with Vivek Swarup, who is at UCI, and he is a real powerhouse on the hippocampus, as well as single cell analysis. Barb Kaloun is our clinical outreach coordinator, and she engages with patients in studies that I'm not describing, that we do work with iPSC cells, looking at neural precursors, and that work is done in collaboration with Athanasia Panopoulos, who is also a faculty member at Notre Dame and an expert in stem cells. And then finally, Vanya Akarian, who is really world-renowned for his work in nociception. And in collaboration with him, we have developed some tactile assays that can be measured in the KS mouse and reflect some of the symptoms that one sees in patients. With respect to past members, Suhail Alam actually initiated this project, and Arpita and Catherine were technicians who worked with him, and Gabby Kim and Vi are undergraduates. I'd also like to make a plug for Chris Patska. He is a new faculty member at the center. He only started his work this year, but he's extremely well trained. He comes to us from Tom Sudoff's lab at Stanford and was part of the team that developed the induced neuron technology, which I know is being used by many labs in Kabuki syndrome. And Chris is also taking that approach, but he has an awful lot of tricks up his sleeve. He's a very smart guy, and I think will make a great contribution. Finally, I would like to thank, in my last slide, Hans Bjornsen. He's done terrific work in the mouse model in Kabuki syndrome, and actually, had he not published his 2014 paper, we probably wouldn't have taken the path that we did. I also had the pleasure to meet with Hans via Zoom one-on-one earlier this year, and we had a really productive conversation. He was very helpful in a lot of different ways in terms of optimizing our assays for KS, etc. And I really think that the idea for the Kabuki Syndrome Foundation promoting collaboration between investigators is a really great strategy. And I'd like to thank the Kabuki Syndrome Foundation. It's been a real honor to work with you, to serve on your medical advisory board. Thank you very much for inviting me to do this presentation. And it's been a great conference. So I really credit you. It's awfully hard to do these virtual conferences, and you've done a terrific job. And Janet, I'm sorry that I've been a bit of a pest. I hope most of the other people were not as troublesome as I was. But if they were, you just really kept your cool. So congratulations. And I'd also like to give a shout out to Renee and ATK. It's really wonderful to see how well two patient organizations work together in a rare disease, and it really is the formula of success. So with that, I'm done.